All right. I thought Sonic Hedgehog was dense, but no. This is dense. Oh yeah. Especially I think the when people subscribe and it plays the Sonic the Hedgehog. Yeah. I have that really loud. So I can imagine um my eyebrows practically fly off my face. Oh my god. Um but yeah, I, epigenetics was very there's a lot, and there's going to be a lot of really cool stuff. Um but fair warning, also a lot of letters. It's okay. We like letters. We like DNA. We like little twisty guys, little stringy twisty guys that kind of look a little bit like jizz if you isolate them, to be honest. um that's okay. Then then it'll be perfect because you don't have to read the letters, you just have to look at the letters. You just have to be like, "Yep, boy are those letters." Uh and a lot of the figures I use are from this place called biorender.com. Um so anything that is really well drawn and cartoony, that is from them. Uh Please don't come after me, biorender. I don't make a profit, I swear. I sound like your second grade teacher. Was she hot at least? Don't worry, Gray. It's okay if you can't read. Pat, pat, pat. You just have to know that they're letters. That's all you need to know. If you look at them long enough, you'll learn to read. Oh shit. Oh shit. That didn't really work out for me when it came to math. But that's okay. I work with a lot of letters. It's mostly overwhelmingly letters, so it's fine. Um in order for me to talk about epigenetics, of course, we need to lay a little bit of a foundation because, well, as epigenetics is very fascinating. A lot of people in the pseudoscience community really like it, of course. A lot of people who want to believe that if they drink like shit tons of grape soda while they're pregnant, their kid will come out loving grape soda, or maybe just will become grape soda in utero. But we're gonna start with just the word itself because we like words. Math cripples my brain too, so we just like words. We'll start with the word epigenetics. Um, uh, Genetics, of course. Oops, here we go. Genetics, of course, is the study of genes. It's the study of heredity, and genes are colloquially just known as units of heredity. Um, what they actually are specific groups of nucleotides on a sequence of DNA. That's the molecular definition that we're going to be working with here. Um, and they're distinct areas on long stretches of DNA that are all organized into chromosomes. Now, epi then means outside of or above. So we'll be discussing concepts that are above genetics, above the code. And it has to do with the inheritance of observable traits that are controlled by more the regulation of those genes rather than like the genes themselves, rather than the code itself. Because again, we're above the code. Yes, your epigenetics for real gave you ADHD. Something happened while you were in the uterus or probably in early childhood that was like, let me turn off all of these genes that are going to help her concentrate ever. Oh my god. That shit's real. That fucking shit's real. Um, so the code. Epigenetics has nothing to do with the code, and yet we must we must understand the code. We must know what's going on at that level if we want to understand the epigenetic mechanisms that I'm gonna go into. Um, I will lay down a foundation by sort of going into genetics proper for a little bit, which is my favorite thing. So, DNA, code. I pretty much use those things interchangeably. It's really easy to just think about DNA as blueprints for things that our body needs in order to function, things that are needed to both create and maintain human life. Um, the most basic unit of our DNA code is the nucleotide, which is pictured here. It is a phosphate group, negatively charged, remember that, attached to a sugar, attached to a nitrogenous base. Now, the bases are what make up the DNA alphabets. We got our A's, our T's, our G's, and our C's. That is a G right there. The nitrogenous base, nitrogenous base right there is going to be guanine. And their specific order and their organization along those long stretches of DNA, aka chromosomes, are what make up genes. And I just imagine, bazillions of these letters. I wish I could, let's see, what's like a really big chromosome? The X chromosome is really big. The X chromosome, I want to say, is like 160 million base pairs. Base pairs are basically what's pictured here. You see A is always with T, G is always with C. 160 million of those, just like boom, 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 boom. That's just one of our chromosomes. I believe that's how big the X chromosome is. So they're all lined up on those two complementary strands that twirl around each other into a double helix. They bind to each other with hydrogen bonds. So in between here is where the hydrogen bonds are, and it's amazing. Literally just the order of these things right here can tell us whether we're at the beginning of the gene, whether we're at the body of a gene, or whether we're at the end of it. It's amazing. So genes are organized, like I said, on chromosomes. This is what they look like when they're super condensed and coiled up. This is actually a duplicated chromosome. Chromosomes are just the one side can be considered a chromosome, but as our cells are always replicating, it's easier to see them when they look like this. So the long strings of DNA that are coiled up, if we unwind them a little bit, it kind of looks like this, and that's where we can see the specific bases and how they interact with each other. We have, importantly, we have 23 pairs of chromosomes. You get 23 from your mom, 23 from your other parent person, and that makes 46 total. So pretty much all of your cells are going to have these 46 chromosomes. Foreshadowing. How is it that every single cell in our body has the exact same genetic code, but they all look so freaking different? Well, we'll just see about that. Epigenetic go brrr. I really wanted to find like a lot more like conclusive epigenetic info on like mental health disorders, but it's really hard because most of it I would read it and I'd be like, this is circumstantial. Like, we don't really know if that's how this person developed this mental disease. And it's also really hard to like pinpoint exact genes. Although I was able to do that for some of them. They should. Okay, but if they don't give you all of what they have, you would just be nothing. And I like that you exist as you. if you only had, I don't know, you need all of them. You need all 23 of these chromosomes, even the ones that are. I think the chromosome 22 is the tiniest one. It's like it's a little fart, but you still need it. You still need all of them. So yes, chromosomes. Very important in our discussion of epigenetics because it's the structure of the chromosome and the proteins that make it up that are subject to these epigenetic changes. I don't know, Amy. I feel like the combination of the two really worked with you.
That's true.
Yeah. You got some gay on your mom's side too, just saying. Okay. So the code. What do we do with it? There's two main things that we do with our code. The first thing is we frickin' make proteins, all right? That's how we're surviving right now. Proteins are any phenotype that you have, whatever it is, hair color, eye color, whether you do this with your hands or this with your hands, or if you can do the that thing, it's because of proteins. So the first thing that we do with our code is make proteins, and let me introduce you then to the central dogma of molecular biology. I did not name it that for dramatic effect. That's what it's called. I think it was one of those Watson Crick guys. I'm assuming the one that Jeff Goldblum played in a movie. I think it was probably that guy that did this. But it's amazing. It shows us the relationship between the string of letters in our body and observable traits that we can see in one another. And super simple. It's 5 words and 2 arrows. We have our DNA. Our DNA gets transcribed into another nucleic acid called RNA. That RNA acts as a middleman and gets translated into a protein. It's not it's one of those things that's really complicated, but it's just not complicated at all. I'm gonna go through it quick and dirty. So don't you worry your pretty little head. First question is probably is RNA like the dollar store DNA? Oh man. Oh, don't tell my RNA that you said that. I love that, but no. And I'll tell you why. We love our RNA. It is not a discounted version of DNA. I would argue that evolutionarily, way earlier in the line, we could say that because technically, there's the theory of like the RNA world where pretty much all life, way billions of years ago before us, was all RNA. And we evolved to have DNA because it's better. Yes, you do. You have RNA floating around in your body at all times. Sometimes not even in this form. I'll show you some other RNAs. There's multiple RNAs. But the reason why we have this middleman RNA, and the reason why I don't want to be too mean and call it dollar store, is because it's doing us a big, huge favor right now, right? Because DNA is in the nucleus, right? And we have it there because it's really well protected. And that's also why we have it in 2 strands. We have basically 2 versions of our code that are super protected in the nucleus, all super coiled into chromosomes, because our code is very valuable and we don't want anything to damage it. However, we need to access it a lot in order to make these proteins. So, instead of just making DNA go out into the cytoplasm, into the cytosol to be translated by the ribosome, where, it's dangerous and there could be some degrading enzymes in there, Instead, we have developed a way to just, the machinery comes to us, it comes to the DNA in the nucleus, transcribes it into RNA, and then the RNA is what will go out into the cytosol, brave the scary environment, and get translated into the proteins. And it's, I mean, that's huge. That's a huge evolutionary thing that we did there. Keeping our DNA in the nucleus, never having to, take it out of there really, and then the RNA can go and brave the scary environment. So really, really cool stuff. Really important. So step one of that is transcription. Again, just gonna go through it quick and dirty. We're gonna pretend that this here is our gene. Our DNA is so sensitive. Like, and you're gonna see how sensitive. I'm gonna go into a little bit about how our code can get a little effed up, like naturally, like not even with radiation or anything, just like it fucks itself up. Truly, storage and transmission of info. Like another way that humans are very much like computers, not just what is it, the electricity fucking in our brains and shit, but how we store our info is just so, it's very efficient. But so to transcribe really quick, just to so that we can make a protein, this is our little tiny, super tiny gene right here. It's only 9 amino acids long. That's 9 BP, 9 base pairs. It's nothing. Only one strand is going to get transcribed into RNA. And it's the same strand every single time. Maybe I'll explain it later if I remember. But we're going to take our one strand and we're going to have some proteins come in and add nucleotides to our RNA molecule, and it's going to look something like this. I know my base pair alphabets, we all do, so even looking at this, you can probably just tell what the RNA strand is going to look like. The only difference, of course, as you can see, is we don't have T in RNA. We have U. And don't worry about that. Don't worry about that right now. The important part is that DNA does not have U. Like 100% does not have it. And it's going to become, oh, thank you. It's going to become important when it comes to our epigenetic discussion about error control when things go shitty. But anyway, this is what our RNA is going to look like. And we call it mRNA, our messenger RNA. It's going to relay the message from the DNA to the machinery that's going to make our protein. So now our brave little RNA is going to leave the nucleus and go to the ribosome. It's a hamburger. It's a little guy that goes meow onto the frickin', it really looks like that. It's so cool. And it's going to read the code that's on the RNA. And how it does that is we have a little, I wanted to think of a word other than code, but it's just a code. It's a gosh darn code. There are triplets of amino acids. There are triplets of nucleotides, that are associated with amino acids. So we have our 9 nucleotide gene, which means our little tiny little, honestly kind of chode of a protein is going to be 3 amino acids long. And there's actually, Gray, you asked about RNA molecules floating around in you. Not only are these messenger RNAs constantly being made, but there's another RNA called tRNA. And it, whoa, I don't know why it looks like that. These are little RNAs that more or less look like that. And on one end of them, they are carrying an amino acid, which strings of amino acids are what compose proteins. Proteins are much like DNA is made of strings of nucleotides. Proteins are made of strings of amino acids. So on one end of this, it's carrying an amino acid. And on the other end of it, it has a little tiny little 3 nucleotide RNA sequence that is going to correspond with the triplets on our mRNA. I would love to do I could do a whole lecture just on the dogma. I could do a whole lecture on just translation and how there's like the tRNAs like do a little dance and they get pulled along this thing and it's super adorable. But it would take up too much time. So that's okay. We'll just look at our codes here. Now, this is a cheat sheet. This is cheating. Because the triplets of amino acids, this is what's going to tell me what amino acid is going to be associated with. So UAC, super easy. First base, U. Second base, A. Third base, C. UAC, it should be methionine. Where are you, methionine? Methionine, where are you? Ha! There it is. This, you know, you don't even need to think about it. You can just look at this cheat sheet, and it'll tell us that the first one is a methionine, the second one is a serine, and the third one's a proline. How do I know that? Because of the chart. How does our body know that? I don't know. Okay, I don't know who invented this, but this is how proteins are made. This is constantly going on in your body at all times. When your body needs something to be made, then this is the process that it adopts. I mean, it's amazing. This is how just the strings of letter get turned into things like insulin and hemoglobin and what the heck is that? Is that a proton pump? No, that's the serotonin receptor. Serotonin receptors, proton pumps, and that's Sonic Hedgehog. Any signaling molecule, any phenotype that you have. It's overwhelming. This process gets adopted, and your body just does it. It just happens. It hurts me. My genes got to third base. Oh my god. Some genes don't make it, you know, the stop codons. That just ends it all. It ends it all right there. So yeah, that's the first thing that we do with our code is we make these proteins. And of course, this reading of the code and pretty much this entire process is subject to epigenetic changes that can alter the expression of these genes, again, without doing anything to the code itself. The other thing that we do with our code, also something that we're just constantly doing into infinity all the time, is replicating our DNA. Anytime your cells need to divide, like right now, I just probably broke a ton of freaking skin cells, and now they need to divide to fix themselves or whatever the heck. But it's happening all the time. We're constantly...
I love
it. Oh my gosh, I do love it. Thank you, Zippy the Deadhead. What's up? We are, you know, constantly growing and constantly being alive, and therefore constantly opening up our genome and just replicating the whole darn thing. And the interesting thing about that is, you know, I think when we think about epigenetics, there's a lot of considering how the changes affect like in more in like a disease sense, like how the changes can affect mental health or memory and stuff like that. And those are real, obviously, but the point that I'm going to be driving home at least for the next probably half hour is that there are epigenetic changes and mechanisms that are normal and that are supposed to just be there. And we need them to be there so that, for example, our bone cells are different from our skin cells. And I'll tell you what I mean in a sec. So let's just, most of us probably went to high school or middle school, so we understand. We know that mitosis is when you make more cells. So I don't need to go into that too much, but I just want to make sure that I mention that the cells at the end of mitosis are going to have the exact same amount of genetic material as the original cell. That is important. All of the DNA is going to get replicated. And in during that replication, it's not just making sure that the code is the exact same or that the number of chromosomes is the exact same, but making sure that whatever epigenetic changes were already present in that cell also need to be maintained through every single generation of that cell type. Um, so, yes, okay, I do mention meiosis. Cool. So, mitosis, same amount of genetic material, important. Meiosis obviously is a little different. Meiosis is when you make sperms and eggs, when you make the gametes. And it's a little different because at the end, you're going to have 4, maybe, different cells that are not going to have the same amount of chromosomes as the parent cell. They're going to be what's called haploid. They're going to have half the amount of genes as the parent cell, which is what you want because that's how we procreate. You want half and half. You don't want that would be too many chromosomes if we had like 46 and 46. What's 46 from 46? 92? No. 92. Is it 92? What's 46 plus 46?
46 plus 46?
That's 92. Oh, are cells gay? They are super gay. Look at this. Look at how. I mean, homo means same, right? Look at the homos down there. Same amount of genetic material. Meiosis is a little. There could be an argument for meiotic cells being gay, though. I'm sure there are. So again, there's still in meiosis we have that sort of DNA replication happening, and importantly, the code is not being altered. We like that. We like it when the code doesn't get altered. Let's touch on heredity just really quick with the definition of a gene. I know it seems simple, but it's a little more complicated because what I mostly refer to is the molecular gene, as you see here. Specific DNA sequence. It is good because it's specific, but it's a little more complicated. It's a little more abstract and maybe hard to consider. But it is more updated because the guy who is considered the father of genetics, Mendel, I mean, he old AF. Like, his discoveries are still important today, but what he considered a gene was not the molecular gene. Anytime you hear the terms like the eye color gene or the cleft chin gene, that's a Mendelian gene. You know, like it's less specific and it is easier to understand. I don't think that it's necessarily wrong, of course, but it's just really reductive. Because take eye color. Eye color is controlled by like 16 different genes. And some of those genes don't have anything to do with skin or eye coloration at all. They're like, they are seemingly like super random. So when we reduce things to the Mendelian gene, it's not bad because it makes it a lot easier to understand. But I will, when I'm talking about a gene, I'm talking about the molecules. That's what I'm mainly going into. So, do I talk about mutation? Yeah, so the other consideration is that much like Darwin didn't really know anything about molecules and genetics, so evolutionary concepts a lot of times need to be updated as we understand more about molecular mechanisms. The same can be said for the father of genetics. Some of his ideas are not wrong, they just need to be updated a little bit because he doesn't know shit about epigenetics. This motherfucker doesn't know anything about epigenetics. Any mutations that happen in the code or that we induce, or traits that are inherited 100% through just the DNA sequence, that's all going to be genetics. But we're above that. We are so far above genetics, but it's still important for you to understand the structure of the code and how and why it's accessed and replicated and read and transmitted in order to see how epigenetic influence affects our gene expression. Um, so now that you've been sufficiently worn the hell down by our genetic code, we can finally go into our epigenetic code. The E code, as I like to call it. E.
E.
Has two main mechanisms that drive it: histone modification and DNA methylation. By the power of these two things combined, we can take trillions of cells in our body, trillions of cells in our body, that all have an identical genetic code and make them different from each other. Cells all have. Again, I just get overwhelmed by this stuff sometimes because it's kind of an existential nightmare. But when I think about epigenetics, I think about how important it is that those concepts are keeping my skin cells from doing weird things like secreting gastric juices or stuff that should only be reserved for a GI cell. We have epigenetics to thank for that. Oh my god. So we're ditching the ATGCs. We're done with the code. We're just gonna look at changes in the structure of DNA and its interactions with the surrounding environment, and the signals that it can get about the genes without affecting the code itself. So we will start, of course, by looking a little bit at the anatomy of a chromosome, because that is DNA in its superior form, of course. It's organized form, even. So these amorphous blobs are carefully crafted combinations of DNA and the proteins that organize it. The DNA plus the proteins is referred to as chromatin. Now, chromatin that is more open and easily accessible and has a lot of gene transcription and gene activity going on, we refer to that as euchromatin. And if we have the opposite, where we have, I don't know why it does this, really, really condensed chromatin that is not very easily accessed and that has more gene repression, that's what we're going to call heterochromatin. What you can see pictured here, these, the string with the little cylinders here, these are nucleosomes. A nucleosome refers to a pretty specific amount of DNA wrapped around a complex of proteins called histones. Just that whole thing, we just refer to it as a nucleosome. And you can see in euchromatin, they're spaced a little bit farther apart. Whereas in heterochromatin, you can kind of see they're super, super close together. Now, like I said, nucleosomes are the DNA wrapped around histones. And excuse me. Let me go back here actually. The genes that are this string is DNA, of course. The genes that are in here, like they're like kind of physically trapped within the histones within the nucleosome. Those genes are going to be a lot harder to access. Okay, so um, histones control chromatin structure. Um, they are what permit the relaxing and the tightening of certain areas of DNA. They allow certain genes to be more active and they allow certain genes to be more repressed. Um, so they have drastic effects on chromatin remodeling, which is, of course, the changing around of the chromatin structure. This can be seen, a type of chromatin remodeling would, an example would be if we have a chromatin remodeling protein, denoted by that little yellow guy. Chromatin remodeling proteins can bind to certain nucleosomes, and they can sort of relax the bonds between the histones and the DNA, resulting in, oh my god, I don't know why it does that, resulting in certain, like an unwinding, or it's called nucleosome sliding, the sliding of the nucleosome, so that, I know it's hard to see because it's just really compressed for some reason, but if this nucleosome moves slightly to the right, you're exposing the genes right here, basically. So nucleosome sliding is an example of chromatin remodeling. Now, I probably don't even need to explain to you the implication of having the ability to keep our genetic code the same, but maybe making it so that certain genes are inaccessible in some cells but not in other cells. Histones love BDSM.
What?
They do. Look at them. They love just being like wrapped up.
Oh.
They love being tied down,
I can
slid
see it.
around.
One's even got a tail.
Yes. The furries. The histones are furries, confirmed. Um, so yeah, nucleosome sliding. What's more, histones are actually complexes of 8 proteins. You don't need to worry about what the proteins are called or anything, but it's important to know that in addition to having the nucleosome sort of slide and change which genes are being revealed, those chromatin remodeling proteins can also just take the whole histone complex out, or they can switch around the order of the proteins, or they can put like completely different histone variants in. I mean, there's a lot that can go on in order to change how the DNA is being sort of wrapped around these proteins, um, changing how easy it is to access certain genes. Um, and yeah, chromatin remodeling proteins are really fascinating and interesting. Why do we freaking need to do this though? I mean, we have the code, right? Why do we need to make all of these alterations to the environment? Well, like I said before, we want to make sure that the right genes are expressed in a cell at the right times. Let's take, once again, mitosis, for example. During interphase, that's when the cell is growing and that's when it is actively replicating our DNA. If we, okay, this is like a fake example because I don't know the genetic intricacies of a bone cell versus a liver cell, but the concept in itself is true. Obviously, those are different cells, right? So, if liver cells are replicating and bone cells are replicating, in addition to copying the DNA code, our body wants to make sure that the right genes are repressed, so it keeps these epigenetic patterns that you will see a bit more of um, in the subsequent generations. You don't want to see a sort of de-differentiation, basically. You want your tissues and your populations of specialized cells to stay that way. So that's why when they mitotically divide, they all of the daughter cells need to have all of the same genes repressed and all of the same genes allowed to be activated. Um, there are some genes that are ubiquitously expressed. They're active in pretty much every cell type. We call those housekeeping genes. Um, and they're usually things that are important for cell metabolism, like things that any cell would probably want to have going on for it. Um, and these are just two examples of proteins that of chromatin remodeling proteins that make it so that the exact correct spacing of those nucleosomes is the exact same in all of the daughter cells after mitosis. Who knew that we needed that? So, yeah, just to reiterate, the genetic code in your cells might be identical, but the code is going to be different depending on the cell type. And different cell types are going to express and repress different genes to fit their own specialized needs. I can't stop thinking about skin cells secreting gastric acid. I really don't like that I said that. So gross. Okay. So beyond uh, the chromatin remodeling proteins that affect, you know, nucleosome spacing, um, we can actually make modifications to the histones themselves in order to give out signals that can say things like, don't transcribe this gene, or please transcribe this gene, or I'm damaged, help. Um, I'm going to talk about just two different modifications that we make to the tails of histones, epigenetic modifications, um, as a way to change the way a gene gets expressed. So, depending on the type of modification, it can increase or decrease the expression. Um, it can signal damage. I said that. Oh, and it can. This is kind of cool. It can pinpoint parts of the genome that were already replicated, so that they don't get re-replicated, and thus um, make it so that daughter cells have too much genetic material, because you don't want that. So the two types of mods are going to be methylation and acetylation. So histone methylation. It is usually associated with the repression of genes, but not always, as we're going to see. Let's see. Okay. Sorry. Histone methylation, you're going to see it doesn't always repress gene transcription, but usually it does, and that's either by the physical, like, chemistry, like if it changes the chemistry of the surrounding area to make the gene less accessible, um, or if it just recruits other proteins to the area that sort of prevent transcription initiation factors from binding. Um, so, usually when we add a methyl group to a histone tail, which are the string of amino acids, we usually add it to the lysine of that histone tail. You can add up to 3 methyl groups, and the number of methyl groups is gonna say something different to the surrounding proteins. For example, let's go over the nomenclature first. Don't worry. You saw earlier, I'll go back really quick. We have H2A, H2B, H3, H4. Yay, those are the histones. If we are on histone H3, and we are on the 9th lysine of that chain, K stands for lysine. I don't know why, it just does. And we add 1 methyl group to it, that's how we refer to that modification. We name modifications in this manner. So H3K9 methylated once is interestingly associated with increases in transcriptional activity. And it's usually in the promoters of genes at the very beginning. So it's possible that for whatever reason, adding this 1 methyl group to that area makes it easier for the transcription initiation factors to bind that area and transcribe and make the protein. However, if we add just 2 more methyl groups, the story changes a little bit. We're doing the opposite now. We are actually making heterochromatin. We're making it way harder to access whatever gene that this is occurring in. And what happens basically is the 3 methyl groups attract a protein called HP1, which is a protein that basically like makes heterochromatin. So for whatever reason, 1 methyl group in that area, totally fine. We can transcribe genes, no problem. Add 2 more methyl groups and make it trimethylated. Now we're recruiting the big guy, the frickin' heterochromatin making protein, and we are condensing our genetic material and making it so that we can't transcribe those genes, protecting them. Dude, hello, how you doing? I'm glad you're here. Hell yeah. Okay. Those are the main things, honestly, that I think about when I'm thinking about histone modifications, is just, you know, increasing or decreasing gene transcription. But there is that other thing about DNA damage, where modifying the tails of a histone can help to repair that damage. So there are, of course, there are ways in which DNA gets damaged that are like exogenous factors like radiation or dangerous chemicals, but also our body does it on purpose during meiosis, which, remember, that's when egg and sperm are made. And during metaphase 1, when the whole duplicated chromosomes are lining up together, they're recombining, they're exchanging genetic material to increase the genetic variation. And when they do that, double-stranded DNA breaks are made intentionally, and they must be repaired so that they can continue to be normal, storage units of our genetic material. And I believe it's the same modification that I was just talking about. Let me look at my notes. That also, yes, it is. Wait. Yeah, H3K9, the same one. So this chart right here when our DNA is being normal, um, then H3K9 has two methyl groups, and it's pretty much chilling. When the double-stranded break occurs, then it gets methylated a third time. So that signals to the surrounding protein machinery that there's a double-stranded break that needs to be fixed. And I won't go into the phosphorylation that takes place here, but I'll just say that the proteins that get recruited to the area um, will actually end up making it more accessible. So, despite the fact that the trimethylated under normal circumstances is something that makes heterochromatin and makes genes less accessible, in the event of a double stranded break, it actually does something different. It recruits machinery that will open the area and make it easier to get to so that the machinery can add base pairs and repair the break. It's how does it know? We just don't know. In case anyone noticed here, this is the BRCA gene that I know. I actually get quite a few questions about that. It's a DNA damage repair protein. So a lot of proteins that are implicated in cancer tend to have that function. So that is that. Acetylation. I just kind of mentioned it, but when you acetylate lysines, it does. I think it pretty much always opens the chromatin structure to make it more accessible. And in this case, I think there is an aspect of like acetylated lysines might recruit proteins that help with the replication or transcription, but it's mostly the chemical changes that it's making to the area, because remember that phosphate group on DNA, it's negatively charged, and the uh, lysine, the end of a lysine on a histone tail is positively charged. Opposites attract, so generally those two are pretty tightly bound. But if you add an acetyl group to that lysine, it deletes the positive charge and it relaxes it chemically without really needing to ask other proteins to do anything. Yes! I knew, I suspected it was you because I think I asked Amy. I was like, that's Chloe, right? It's gotta be Chloe. I appreciate so much that you remembered. Freaking victory. Ah, I need lemonade. Lemonade is necessary. Why so good? All right. So, I know there were a lot of odd words that we don't hear in our everyday life, so I am just going to take a second to summarize some things. Our DNA code is ATGCs. Our epigenetic code is H3K9. It's modifications to histones. It's changing how the DNA is interacting with the surrounding proteins by either changing the charge, like with acetylation, taking away the positive charge and making it more relaxed, or recruiting other proteins. Uh, and those proteins can either act to further condense the genetic material into heterochromatin, like HP1, Or they can do the opposite. They can increase the accessibility. Um, these are all ways for us to remodel chromatin through the histones themselves, through the core members of the nucleosome, that of which there are a lot of that compose the chromosomes. Um, Histone modifications are important not just because we want to keep our cells differentiated, but because we want our genome itself to be maintained. Um, Earlier, I talked about the reason why we have an RNA intermediary during transcription and why we don't just like do it straight off of the DNA. It's because our DNA code is so goddamn valuable. And these histone modifications ensure that the integrity of that code gets maintained um, through generations of divisions and replications and anything of the sort. And this, what I just said is, fuck you, broke bitch. Body was like, I'm not replicating anymore, and now all of your cells just have like 2 strands of DNA. My body only has my stupid genes replicating and transcribing now. All my smart genes methylated to hell. Inaccessible. Can't do it. This this concept though, this maintenance of our genome that's aging. As we age, our body starts to kind of lose this ability to maintain the integrity of our genetic code. And that's why a lot of the more dangerous effects of aging tend to be just very clear signs that our body, oh my god, hair, that our body is not making itself like it used to be. So this figure right here, this amazing figure, shows what happens when our DNA is damaged and why our body's own process kind of contributes to the aging process. So up here we have that H3K9. We have this normal methylation pattern right here. When it experiences a double-stranded break, a enzyme comes in to repair the break, and when it does that, it actually changes the epigenetic pattern. Instead of just having 2 nucleosomes next to each other that have 1 methyl group each, now those methyl groups are spaced apart by 2 nucleosomes that have acetyl groups. I mean, that's a huge change. But that's, that's one of the normal mechanisms for repairing a break. So if that occurs many, 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 many times throughout your lifetime, that's how we get these effects of aging that we see. I forget what this is, but this is, I believe, nerve cells, which is depicting the like neurodegenerativeness of, you know, inevitable aging. Um, Hell yeah, dude. Yeah, your body is gonna continue being so good at making itself and like dividing and just like, you know, you just have minimal fuck ups in your cell. Your grandmother has minimal fuck ups in her cell division. Um, so lucky you. I'm just kidding. Oh my god. Yeah, it is like it's really scary. It's the lack of control. Okay, but here's the bright side, sort of. So this is the epigenetic change that we saw that, accumulating this change leads to aging. Um this lab added these factors that remodeled the histone pattern back to what it was before. And when they did that, I don't know if it was like terribly conclusive for them or not, but I think that in their mouse model, they did see some like reversal of certain like aging effects. So, um this accumulation of epigenetic changes as a result of regular run-of-the-mill DNA damage um is is very fascinating. How it plays a role in getting old. I'll bet aging is a hot topic. Aging is
Yeah.
really up there.
Most well funded by the world's oldest freaks with lots of money.
Ah! I mean, I think everyone wants to stop aging, and people are really into like there's a book called Beggars in Spain, and it's about genetically engineering people so that they don't need sleep. Um and those people, the sleepless, they like didn't age and they lived a really long time. It was fun. Okay. So, yeah, cool. We can modify histones. Um but what's that other thing we can do? Oh yeah, we can methylate the DNA, but not acetylate, just methylate. um just like with histone uh methylation, it's pretty much almost always associated with the repression of genes. And it is absolutely required for cell differentiation and embryonic development, the formation of memories, learning, and exon activation, which we will talk about in a little bit. Um, so a gene is pretty much always going to be repressed if its promoter is methylated. A promoter is the very beginning of the gene. That's where transcription is first initiated. So if the protein machinery cannot get to that area, no transcription. Um, so yeah, the methyl group itself is physically impeding the binding of those proteins. But again, the methyl group can also just recruit other proteins to act as like bouncers for the transcription initiation factors and be like, get the hell out of here. That's how they talk. They talk.
Wow.
I'll allow it. They give you a shit pair of kidneys. Oh no. Oh yeah, you I know you have to pee a lot. Oh man. Come on, Gray's Genetics. Give better kidneys. Make her pee once a day. Actually, that might not be good either. Who knows? Okay. So yeah, I think it's safe to just assume DNA methylation, gene repression. Hello. There we go. Just real quick, just some chemistry, super fast. All right. So pretty much the only base that gets methylated is C-cytosine, which looks like that. I think adenine sometimes gets methylated, but it's always cytosine. We just care about cytosine. And when it gets methylated, it's pretty much always on the 5th carbon to make 5-methylcytosine. It happens to be in the exact same position as thymines. methyl group. Um, so deamination is the process by which amino groups are removed, and it just happens as normal pyrimidine metabolism. It just happens. It's spontaneous. Um, Now, normally, um, if regular run-of-the-mill cytosine gets deaminated, it turns into uracil. What? But that's fine because uracil is not supposed to be in DNA, and so DNA repair enzymes can recognize that immediately and be like, "What the hell?" and just take it out and boom, replace it with the correct base. Error control. We like that. The fact that uracil is not in DNA and the fact that our body can recognize that can get these problems fixed, no problem. However, if 5-methyl cytosine gets deaminated, which it definitely will, it turns into thymine. See? You're deleting that, replacing it with a carboxyl group just like over here. And like that's a lot harder for our body to sort of recognize in terms of thymine belongs in DNA. It's supposed to be there. So instead of coming in, remember that C goes with G. So our body sees a TG, and it's like a mismatch repair. All I gotta do is put the right base in there. But instead, wait a minute. Yes. Instead of the body seeing the mismatch and being like, oh, this T was originally a cytosine or a 5-methyl cytosine, I'm gonna put a cytosine in there. That's too much. The machinery doesn't go that far. It just sees a mismatch. So it might just take the G out and switch it for an A. Yay! Now, haha, it's fixed because A goes with T, except no, because that's a mutation. That's literally just a mutation. So interestingly, even though we have a lot of these 5-methyl cytosines in our genome, because that's how we regulate our genome epigenetically, it does increase the chances of mutations like that happening just for that reason, just because of that deamination into thymine instead of uracil. It makes it really hard for our body to figure out that a mistake was made. Oh my gosh. Yeah, holding your pee is really bad. I did that a little bit as a kid because I was just nervous about using bathrooms, but luckily I didn't like maintain that very long. I just like was like, just deal with it, just use the bathroom. But yeah, that'll hurt you. That'll hurt your kittle knees. That's a lot because I feel like I probably pee like maybe 3 times a day, maybe like half that. Amy pees every 5 seconds. Every 5 seconds. Just last night when we were trying to play games, after every game, it was like, hold on, I gotta go pee. Hold on, I gotta go pee. Hold on, I gotta go pee. I'm getting for the millionth time.
I've got the world working against me when it comes to my piss refractory period.
She drinks a lot of water. She's healthy, so that's good.
Yeah, I drink a lot of fucking water.
Oh, you say, yeah, 8 to 10 times. Holy shit. Is this how cancer starts? Yes. Yeah. That gene I mentioned earlier, the BRCA gene, which is one of the breast cancer genes. Don't quote me on this. Mutations, there's different types of mutations, but if there was something where there was a 5-methylcytosine somewhere in that gene, and it changed, and there was the mismatch repair that went wrong, you could potentially make it so that that gene doesn't work. And it's a tumor suppressor gene, and you want your tumor suppressor genes to be doing their job. So if a gene like that were to have a mutation, where even just the one genotype, one nucleotide, which is a single nucleotide polymorphism, if that made it so that it didn't work, then yeah, totally, that could be boom, cancer. Um, Deamination, like amino acid, it is taking off the amino group. So basically.
Okay.
And yes, oh my god, yes, Spyro will make you piss forever and ever and ever because it I think it impairs aldosterone. Hold on, I have to think about this. Aldosterone makes you take in salt. And when you take in lots of salt, you retain a lot of your water. Yes. So you're retaining less water, you are pissing it out instead of taking it in, I think. Yeah. Fun, aldosterone.
I fear for the day where I'm finally off of that medication and my body no longer flushes salt all the time, and then I have to cut back on all the really good, yummy, salty foods that I eat nonstop.
No, you won't.
I think I will.
No, you won't, because you drink a lot of water. You will. I think you'll be okay.
I'll just like retain water and get all bloaty.
Because I can't imagine you being like, oh no, now I have to eat less pickles. You won't do that. You will never ever do that. You will always want to eat pickles. Ah.
It predates the medications for sure.
Oh yeah. God. But yeah, interestingly, despite the fact that we really need this mechanism where we are methylating our DNA, this is just, this is a fact of life. It happens. It increases the chances of these mutations happening. So DNA methylation itself is seen as the default state. It's just seen as the default state in cells, and there are genes that have to be specifically selected to demethylate and activate. And remember the differentiation, the distinction between the bone cells and the liver cells and the housekeeping genes that are just ubiquitously expressed. In addition to those histone modifications, we of course have DNA methylation in play for these mechanisms. So yeah, cool. Huh. All right. 2 types of DNA methylation. Maintenance, of course, is that's taking place in replication, for example. Just like in mitosis, the nucleosome distances and the histones and all that stuff need to be maintained through the generations of cells. So too does the pattern of DNA methylation. So DNA methyltransferases are enzymes that add methyl groups to DNA, and they preferentially add that to what's called hemimethylated DNA. What that means is one strand is methylated and the other one isn't. So the DNMTs are kind of able to recognize that and then boop, 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 make sure that the other strand also gets methylated. Excuse me. So this would be, that would be maintenance. Once again, maintaining the genome and the epigenetic patterns. De novo methylation is the creation of new methylation patterns. I'm gonna show you. I'm gonna show you real hard de novo methylation during embryonic development, but it is also happening in our bodies right now. hematopoiesis is the differentiation into the different types of blood cells. So, uh, when and those are types of stem cells. They're hematopoietic stem cells. So you can see here, um, this lymphoid progenitor cell is going to become a couple different cells. Same with this myeloid stem cell. So you don't necessarily want to want the daughter cells to have the exact same um, uh, sort of epigenetic patterns. You don't want to do that full-on maintenance methylation because it's not going to stay a myeloid stem cell. It's going to become maybe a neutrophil or a red blood cell. So in this case, where you have cells differentiating, you would see the de novo methylation. You would see changes in the methylation patterns happening in subsequent daughter cells because they're becoming something else. You're not just copying them. So it's gonna happen. We gotta. If you were here for the Sonic Hedgehog lecture, you got absolutely ass blasted with development jargon. And we're gonna do it again. We're gonna we have to, because the main, at least in my mind, one of the most important functions of epigenetics is developing the human embryo. I mean, it's so cool. It's, oh my god, it's one of my favorite things. But I'm gonna give us a little secchi. A little secchi. Speaking of pissing, Amy's gonna go pissing. Man, for my future lectures, I really I want to figure out a way to do a quiz at the end, and like have there be a winner and give a prize to the winner, but I would have to figure out the logistics of that. I actually think, I'm sure there's like a Jackbox. Sometimes they have like little templates and you can like make your own trivia game. So like that would be so cool if I could make something like that, but then I would have to think of a prize to give out. And my go-to is always a Steam game because it's so easy to just like gift someone a Steam game. But yeah, that'd be a really fun. Okay. Development. I'm getting ready. I'm getting ready for the development. Okay. Here we go. Development. We know how it happens. Sperm touches egg, becomes massive unspecified cells, becomes massive sort of specified cells, becomes living, breathing human baby. Tale as old as time, Beauty and the Beast. We know this. There are epigenetic functions that exist during this process that make it so that the right cells are expressing the right genes at the right time. So it's a location thing and it's a time thing. It's nuts, is what it is. Um, more so than just the fetus itself, the cells that are supporting the embryo also need to have the correct epigenetic patterns. So it's really just an orchestra that is so beautiful and complicated and terrifying, because if anything goes wrong, it's really fricked up. Okay, but before I delve into, I just have to make a quick statement. about how I inescapably and correctly need to refer to things as male, or paternal, or female, and maternal. And when I use those words, I'm not referring to gender like at all. Uh, Female and maternal is just referring to the passage of genes through a germline that involves eggs and any individuals that might contribute those genes. And of course, male and paternal would refer to a germline big pass through sperm, composed of sperm, and any individuals who contribute those types of gametes and genes. So that's what I mean by that. Anyway, look at this beautiful. Whoever made this, oh man, I forgot to put their name. It was someone, oh my god, I'll have to find it. It was like Marius Martin or something. Someone made this, and it's amazing. It really, let me take you through it, because it's gonna be fine. This is a mouse, by the way. I will say this is a mouse embryo, but we're like pretty sure this is what happens in humans as well. So we'll look at the axes here. Up here means that there's a lot of methylation. DNA. We're looking at DNA, by the way. A lot of DNA methylation, and therefore a lot of gene repression. As we go down here on the lower part of the y-axis, we're looking at lots of demethylation. So lots of allowing gene activity, allowing them to express themselves. So those are the axes. We're starting pre-fertilization here. So in a regular old sperm, there's a lot more methylation happening versus the egg. I'm not entirely sure why that is. I don't know why like the entire sperm genome is just like methylated. My guess is just sperm complete meiosis, whereas eggs do not. Eggs do not complete meiosis until fertilization. So maybe having more gene activity just means they have more things available when that event is going to take place. I don't know. Either way, they combine to form a zygote, of course. And right after fertilization, we see a mass demethylating here. So the cells continuing to divide. I want to say like right here is like the 16 cell stage, which is the morula. Do I have a picture of the morula? Yes. Morulas have totipotency. They contain the cells that can become literally anything, whether that's a part of the fetus or extra embryonic. We call that totipotent, which makes sense because that's the lowest methylation state that is in our life cycle. Those cells, part of what makes those cells have this really high differentiation potential is the fact that pretty much all their genes are accessible. So that's important because as you can see, as we continue developing, that doesn't last very long. Once we start making the epiblast, which is, I believe, implantation. This is around implantation. So we have our cells that are becoming the fetus, and our cells that are separating a little bit and becoming the like the placenta, the extra embryonic tissue. No longer totipotent. Can't become anything because they're embryonic and extra embryonic, but still pluripotent. They still have a lot of differentiation potential. That's going to get less and less and less and less as we keep going. Once we reach here, we're at pretty high levels of methylation as our cells are beginning to differentiate and become what they're supposed to be throughout the rest of the life cycle. This is also where the cells that become the germline, aka sperm and egg, split off from the body tissues or somatic tissues. So take note that the somatic tissues stay methylated. There's another huge demethylation event that takes place as these primordial germ cells separate from the body tissues, um, and once again increase their differentiation potential. It seems even more so than the morula, like somehow more totipotent, I guess, than when we were at this early cell stage. Um, the the PGCs will eventually become the gametes, and as these cells differentiate again and wait, I think I have, yes, the math demethylation. Now, as these germ cells start to become what they're supposed to be, egg or sperm, they start migrating toward what's called the gonadal ridge. Now, in that area, depending on the genetic makeup of the fetus, we're either going to see the gonads develop, and it's either going to be the testis or the ovary. And depending on the gonads, we're going to have different cell types in here. The testis are going to have Sertoli cells, ovaries going to have granulosa cells. Those cells are going to send signals to the primordial germ cells to help push them along their differentiation journey until they become either egg or sperm. Simple as that. um, so the development of the gonads is a way in which the sex of the fetus is going to affect the fate of the primordial germ cells. There's another concept, though. A concept that really, 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 really pisses me off because I had such a hard time with it and I wasn't even going to talk about it, but now I am. Okay. The sex of the parental chromosomes can uh, can determine certain things about the epigenetic patterns present in the fetus. And it's called genomic imprinting. God, it pisses me off. Earlier, I showed you two demethylation events. One here just after fertilization, and one here when the germline splits from the somatic cells. Imprinted genes will completely ignore the first demethylation event. They'll stay there. They don't care. Oh man. The second demethylation event, though, it does erase the imprints, and yet somehow they get reestablished when the germline fully differentiates. Confusing, right? I was so determined to understand this, and I really didn't get it until I straight up stared at this one figure in my little Bible that I love so goddamn much. I literally took this picture from my physical copy of this frickin' book. Okay. It's the closest it took me to comprehending this. Okay. I'll give a disease example for this because that also really helps. But look at this. So we have the female mouse, right? These are methyl groups, of course. So this allele, or variation of a gene, is methylated and it is inherited from the father. So the paternal allele in this example is always methylated. The maternal allele is never methylated. So, right out the gate, the methylation patterns are different depending on whether the allele is maternal or paternal. But what the hell? How is that even established? Now, development happens. The imprinting gets removed and is instead replaced by the sex specific imprint. He's scratching the chair.
Yeah. Should I go get him?
Nah. If you want.
I can fold the chair up.
Okay. So, sorry. They get their methylation patterns removed because that's what happens at the beginning of germ cell development. And then they get reestablished in a sex specific manner. So, in this example, the maternal imprint is no methylation, and the paternal imprint is all methylation. So, this mouse, all of her gametes, all of her eggies are gonna have that same allele unmethylated, even though she inherited the methylated imprint from her father, from the male mouse, because the mouse is female. It's gonna infix the maternal imprint in all of her gametes. And in the male mouse, all of his gametes are going to have the paternal imprint, which is always methylated. I sound like I hate this, but I love this. No, I do. It just... I went through so much to, like, conceptualize this, and the answer was in my fucking textbook that I had this whole time. Okay, so I like that they use colors, because in the original mice, the paternal allele is orange. Now in this one, it doesn't matter that this was originally from for this mouse, from this mouse's like from the paternal allele, because this mouse is female, so it makes the female imprint. All right, so now the eggs are all, because all of the eggs are unmethylated and all the sperms are methylated. Every single offspring, if they have a methylated allele, it's gonna be from the dad, and if they have an unmethylated allele, it's gonna be from the mom. Pissing me off. I love it, but it's pissing me off. Okay. So, why is that important? Why does that matter at all? Actually, you know what I'm gonna do, you guys? You know what I'm gonna do? I didn't write it down because I hated it. Before I go into the disease example, I'm gonna read for you guys the current accepted scientific reason for why genomic imprinting exists. It's gonna blow your mind and maybe piss you off like it did me. Hold on. It is some weird shit. I have so many tabs open. I'll find it though. The reason why we have different epigenetic patterns based on the sex of the developing bee-boo-boo. Okay. The reason is so funny. It's like a fucking weird evolutionary thing. Please give me... No, give me the development. Okay, now I know where to get it. It's something where I read it where I'm like, is this sexist? I don't know. It could be. It might not be. It could be, but maybe. I don't know. Yes, we are. Hypothesis on the origins of imprinting. I couldn't find my scientific article, so this is from the wiki. I'm sorry, but they probably got it from that article, if I'm being honest. Okay. It is the parental conflict hypothesis, also known as the kinship theory of genomic imprinting. This hypothesis states that the inequality between parental genomes due to imprinting is a result of the differing interests of each parent
Mhm.
in terms of the evolutionary fitness of their genes. The father's genes that encode for imprinting gain greater fitness through the success of the offspring at the expense of the mother. The evolutionary the mother's evolutionary imperative is often to conserve resources for her own survival while providing sufficient nourishment to the current and subsequent litters. Accordingly, paternally expressed genes tend to be growth promoting, whereas maternally expressed genes tend to be growth limiting. In support of this hypothesis, genomic imprinting has been found in all placental mammals where post fertilization offspring resource consumption at the expense of the mother is high. Okay, like I don't want to it's not sexist. I was being a little dramatic when I said that. It's not sexist. It's just I there's such a small amount of genes that are imprinted that I would have to do a lot more research in order to determine which ones are growth limiting and which ones are growth promoting. You know what I mean? Like I would really have to see like an imprint doesn't always mean methylated or downregulated. You know, like I could potentially look at genes that are maternally imprinted and maybe they upregulate resource consuming genes. I don't know. Like I'm looking at the citation for this. It's from 1991. I'm not gonna. That's not bad. I get a lot of my stuff from the 90s. They really liked molecular biology in the 90s, but
You got all this stuff from the 90s.
yeah, parental antagonism. I think it's just something that I would personally want to I want to I didn't have time to like really just go hardcore into evolutionary genetics. But like let's look at the disease. Let's look at the disease that I have as an example. I don't know a lot about the genes themselves because if I went into them, maybe we could kind of determine at least if the genes in my disease example were like resource limiting or not. But I like it's just so I want an example. Another hypothesis, coadaptivity to improve fetal development and maternal provisioning. That would kind of make sense. Expressed in the placenta. Because I think I have heard that a lot of these imprinted genes have to do with the placenta, like the extra embryonic stuff, which I'm like, all right, that will make sense. Dude, yeah, it is weird. Most people with shit like that, immune diseases and fibromyalgia, that's another like overwhelmingly women have that disease. And yeah, I wonder what the hell is going on that makes us so much more predisposed to those diseases. Um, because I'm going to talk about another epigenetic mechanism that explains why males are a lot more susceptible to certain genetic diseases. But yeah, what the fuck is? I wonder. This is fake. This is semi-educated guess here. Uh, I'm gonna make that educated guess later. Actually, when it makes more sense. I might have a little bit of an idea. It's probably our bomb ass pussy. It has to be that actually. There's no other explanation. Okay. Oh fuck. So, I did find out through my more research on this that um the methylation of H3K4 and acetylation combination was maintained in unmethylated imprints Whereas for the methylated imprints, there was always this H3K9 trimethylated, H4K20 trimethylated, H2AH4 arginine trimethylated. And my guess on that is like, because those are histones, right? We were talking a lot about DNA methylation, but those are histones, right? So my thought is that even though they're getting the DNA methylations are getting erased right before egg and sperm develop, clearly the histone and the nucleosome, architecture is maintaining these signals that are saying, hey, and they're only on the one parental chromosome, right? They're saying like, hey, maintain this shit, but only on this one chromosome. Um, yeah, fuck this shit. I love it. Okay, so the disease though that is associated with this genomic imprinting concept is um, Angelman syndrome and Prader-Willi syndrome. So, oh, basically, what's happening in Angelman syndrome, both diseases are dealing with the same like cluster of genes, basically, pretty similar cluster of genes. And normally, as for Angelman syndrome, the gene is only expressed, like this is in anyone who doesn't have the disease, it's only expressed from your maternal copy of your maternal allele, and your paternal allele is not expressed. So, people with Angelman syndrome have probably a mutated copy of the maternal one, and they don't have another copy to work with. They don't have, so they not having a maternal copy means no good copy. Same with Prader-Willi, pretty much the exact same concept in similar genes, but with the paternal copy being the one that you want to have functional, and then the maternal copy we just don't use. So, fairly interesting. Yeah, yes. Like I've seen pictures of like children with Angelman syndrome. They just they're always smiling. Yeah.
Really?
Yeah,
Oh,
they
I thought that was a. Oh my god.
no
Is it
that
like cherubism?
not cherubism because they don't have like fat cheeks or anything, but their affect. They always have like a happy affect.
Interesting.
Uh
I've never heard of
yeah. And then uh it's an intellectual disability though.
angels. Oh, really?
Yeah. Same with Prader-Willi, but Prader-Willi, the physical, it's not associated. It's very different. It's not like there's no happy affect. I think it's heavily associated with like obesity. Yeah, very, very different phenotypes. Very, very interesting. But yeah, they be smiling. um,
Wow.
yeah,
Okay, now I'm gonna look it up. Am I gonna?
yeah. It's not gross. It won't scar you like some of the Sonic Hedgehog things to look at.
Yeah.
Okay. All right.
Oh, I see.
Done with genomic
Yeah.
imprinting. Yeah, yeah. See, they're just like they're
Mhm.
smiling, little smiling kids. Your mother gave you obesity. She imprinted. She imprinted the obesity gene. All right. Now we're gonna talk about something that is super badass. X inactivation. X inactivation. It sounds super badass. It's also known as lionization, which is also freaking sounds badass. Named after Mary Lyon, who discovered it, basically. Uh, it is an epigenetic function that we have in order to equalize the amount of genetic material that is expressed in people. Because when it comes to physiological, like males and females, generally males have a lot less genetic material to work with. As you can see right here, the XX karyotype, the X chromosome is huge. 160 million base pairs, 800 genes. 60 genes. That's nothing. So we have this X inactivation to act as a mechanism for dosage compensation. Um, the the dosage is compensated by inactivating one of the X chromosomes in XX individuals. Um, it is always one X chromosome that gets to be active. So even if an individual has Klinefelter, let's say they are XXY, they will still only have one bar body or inactivated X chromosome, or even someone with three X chromosomes, they will have two bar bodies and one X chromosome. You always end up with one, no matter how many X chromosomes you start with. And the inactive X chromosome, it's random, but the inactive X chromosome will remain inactive throughout the rest of the individual's life in that cell. All of the subsequent cells, if it starts inactivated, it's going to be inactivated for the rest of that cell's life. So, a really, really classic example of you wish you got 3X chromosomes. That would be cool, but again, I think the extra chromosome, you could be like the sexiest person. Oh my god, a porn star actually with 3X chromosomes. You would confirm trisomy X equals bomb ass pussy. Um. So, kitty cat, tortoiseshell. Tortoiseshell cats and calico cats are the bestest examples of X inactivation. I took this. This is on the head of the wiki page for X inactivation. I had to use it. Do you see this cat? He's so. I mean, look at him. He's so like, who the fuck is taking my picture? I love this cat. I'm shit. Don't fall. I love this cat so goddamn much. It's like anyway, really good example of X inactivation because the in cats the gene for color, for fur color, one of them is on the X chromosome. And in this case, the alleles for that gene are orange, not orange. That's it. So in calicos and tortoiseshells, you can see how if they are heterozygous, if they have one orange and one not orange, and it's random which X chromosome gets inactivated in which cell, the ones that are active orange will be orange, and the ones who inactivate the orange will be not orange. That's how the cat genetics work, I guess. There's also another gene, I think, that controls the spotting, which is why in tortoiseshells it's a little easier because the black is the not orange, but in calicos, of course, you also have a lot of white. So I think there's another like spotting gene that comes into play there, but in general, this mosaic coloration is because of one X chromosome being just completely inactivated. This process is so weird and amazing. I love it. In some animals, it's always the paternal X chromosome. I think in marsupials that's how it works. But and that would be an example of genomic imprinting. The paternal chromosome is always the one that we're inactivating. But that's not how it works for us humans. It is random, and it occurs at the blastocyst stage. So those cells of the inner cell mass, which are going to become the us, it's once X chromosome inactivation starts, it's going to be super random whether it's the maternal or the paternal one. So let's look at the molecular mechanisms. I got so much of the information that I'm about to tell you about X inactivation. I got a lot of it from this lecturer named Jeannie Lee. I think she lectures at Harvard. She did an amazing presentation on this project, on this process, and I believe a lot of the things that I'm about to tell you were like discovered by her, I think. So and I'm in my when I post my sources, I'm going to post a link to the videos that I watched. She just did such an amazing job. So let's look at this cool ass shit. Um we think that uh of course it starts in the blastocyst stage after we have embedded into the uterine wall. Um there are um there's a lot of questions about this process, but the first one was like, you know, how how do our cells know that something needs to be inactivated? And it was discovered that um our cells our cells count the amount of X chromosomes, sort of. It's more that they determine the ratio of sex chromosomes to autosomes, which are all the other 22 chromosomes. Um, and of course, in males and people who have the XY combo, their ratio is going to be 0.5 because the idea is um, these these chromosomes are making material, so the amount of material in an XY individual that's going to be coming from the X chromosome is going to be half as much as not just of the autosomes, but of individuals who have two X chromosomes, which is denoted right here. So, uh, Dr. Lee visualized this by showing, like, okay, the chromosomes during development, they're making their proteins. As you can see, XX individuals are making a lot more of these specific factors because they have more X chromosomes. And so, our body, that's how our body sort of determines that, all right, something's got to go. There's too much of this shit. Something's got to go. And this partially gets determined because these factors, these circles are factors, are going to bind a section of the X chromosome called the X inactivation center, or XIC. So this is what happens when the factors from the autosomes and the factors from the X chromosome bind to the male X chromosome and prevent it from being inactivating. Because that would suck, right? I mean, that's your only X chromosome. You do not want to inactivate your only X chromosome. So, the body determining the dosage in this manner prevents the single X chromosome from getting inactivated. It's a different story if you have two X chromosomes, of course, because the combination of these factors will bind the X chromosome, but because there's more factors from the X chromosome being made, then it's postulated that some of the X chromosomes are only going to be bound by one of those things. Um, we're going to get into the gene details. This is very over the surface, but what this is basically showing is that there are different amounts of proteins, of course, being made during development, and our body can use this as a way to determine the weight of our genetic material as a whole versus from the X chromosome. Holy shit. All right. So, of course, we should realize that there's definitely a lot more factors, a lot more blue and red circles that go into this than what I'm about to show you. And again, a lot of these concepts, I'm pretty sure, come from Dr. Lee herself. It was a very fascinating video. So, these are some of the genes involved in X inactivation. Don't worry, it's okay. It's fine. These are just denoting the relationships that they have between each other. So, exist or exist is that the main gene that's going to inactivate an X chromosome. Oh, it do. Your chromosome is polka dotted. I know it. It's gotta be. um, and again, with the RNAs just floating around in your body, exist is an RNA. It's not a protein. It is a gene that encodes an RNA. And the RNA has the function. It does not become a protein. Fascinating. The exist RNA coats the X chromosome that is going to be inactivated and causes it to be, methylated, super condensed, boom, heterochromatin bar body. That's what exist does. T6, which is exist backwards. Dr. Lee pronounced it Psi X, but I like T6, so I'm gonna say T6 because you're allowed to just do that. Haha. T6, this little the line with the bar means that it inhibits. So T6 inhibits exist. It makes it so that it does not inactivate a chromosome. And then excite. This is what we do. Excite is an enhancer of T6. CTCF is, and all of these are both RNAs. They are opposite RNAs to each other. And then excite, I don't actually think that excite becomes a protein either, and it might not even become an RNA. I think it might just be a regulatory sequence. So it functions as just the code itself. CTCF, though, is a protein. And it is not made on the X chromosome. All these guys come from the X chromosome. This guy comes from an autosome. This guy comes from somewhere else. And it pretty much inhibits everyone here. All right. I'm going to show you some shit. But I want to make sure that we know. Exist is the main thing that inactivates the chromosome, an X chromosome. T6 inhibits it. That's the big one. So, normally, this is like right before X inactivation. T6 and excite are inhibiting exist. So both of the X chromosomes are active, and they are making their proteins, and they're telling our body the dosage, basically, and letting our body know that there's two of me, one of me's got to go. At the beginning of inactivation, it seems that T6 and excite also recruit CTCF to their sites. CTCF, of course, inhibits T6, but there's this sort of reciprocal thing going on where those two genes on the X chromosome are recruiting the protein to the site. At the very start of inactivation, these chromosomes come together very, very briefly. And this was a huge question that Dr. Lee was trying to answer, and that is like, are the two X chromosomes communicating with each other, and how are they choosing which allele is gonna, get inactivated and which one's gonna be just the active X chromosome? So she explained that they come together very, very briefly, and that the CTCF is a very important part of that connection between the two chromosomes. And then when the chromosomes come apart, all the CTCFs just go with one chromosome. Okay. Okay. And as a result, the X chromosome that does not have any of the CTCF, the T6 and the excite are allowed to keep repressing the exist, and that becomes the active X chromosome. The X chromosome that has all the CTCF is all of that is going to inhibit T6 and excite, allowing exist to coat that chromosome and turn it into frickin' heterochromatin. Save some of that for me. I'm so hungry.
I'm built different.
All right. So, summary: one X chromosome is epigenetically modified such that it is completely inactivated. And it becomes super condensed into heterochromatin. Uh, Diseases that are X-linked, meaning that their diseased genotype, allele, whatever is on the X chromosome, uh, tend to only show up in males, in those who only have one X chromosome. Hemophilia is an example of that. So if and it's recessive. So if uh, an individual with two X chromosomes is heterozygous, they have one normal and one mutant allele on both of their X chromosomes. Um, if they give one of their mutant X chromosomes to any of their XY children, because they only have the one X chromosome, they don't have the other one that has the dominant gene that could potentially mask its effects. Hence, another one will be color blindness. That's why mostly XY individuals are colorblind. Color blindness would be an example where you could definitely have XX individuals who have it because it's not a disease that would prevent you from reproducing necessarily. And there are certainly cases where XX people who are heterozygous can still have like some of the symptoms. I think it's been shown that not every single gene on the inactive X chromosome gets 100% repressed. But generally, that's how it goes. You don't see red flags. X chromosome gave you rose-colored glasses. Oh my god. All right. X inactivation is really cool. I was so happy to find that lecture by Dr. Lee. And she put it in such great terms. But it's time for our last function of epigenetics, which is learning and memory. So when you are learning and you are creating memory traces, this is occurring in your hippocampus, which is where memories are formed but not stored. The formation of those memory traces involves a lot of, events of methylation and demethylation during the process. So, um, your chromosomes need a lobotomy. Off with their heads. um, it's a little hard to conceptualize a memory trace having to do with DNA methylation, but there was a group of scientists kind of made it a little clearer. What they did was they trained rats to be afraid of something, uh, meaning that the behavior that they would exhibit in response to being afraid was that they would freeze. So that's called fear conditioning, and it's a form of learning. What they did in those rats was they gave them DNMT inhibitors. Those are DNA methyltransferase inhibitors. Normally, DNMTs will add methyl groups to DNA during these memory trace events. And they noticed that the rats that they gave the inhibitors to had a lot less freezing behavior. Like, they basically wouldn't respond to the aversive stimulus, which shows that preventing these methylations can inhibit the ability to stabilize memory traces and to learn. I think they were. I think once they were taken off the inhibitors, they were like it wasn't permanent. They were eventually able to learn, the fear conditioning response. Um, but I think another thing that was tested was they they tested the DNMTs in the prefrontal cortex, which is where we have a lot of our cognitive processes like executive function and stuff like that. Um, and the DNMT inhibitors, so making it so that there are no methylation events or lower methylation events, made it more difficult to recall memories that were already there, but it didn't seem to affect memory formation, like making new memories, basically. So it seems that these epigenetic events are really important when it comes to maintaining memories, but maybe not necessarily making new ones. And it looks like potentially methylation and acetylation of histones would also play a role in memory formation. It looks like I don't have studies, but it from what I've read, it seems like acetylating histones is needed when it comes to like
I'm so dumb. Ah, euthanize me.
euphemize me. I think I do say euphemize. It's needed for synaptic plasticity. So, that incurs that's the flexibility that your brain has, and it's also needed for memory consolidation. Whew. So, development, gametogenesis, exon activation, and memory are all epigenetic mechanisms that are just in our body normally and that we need in order to survive. What about them disease implications, though? We love those. We love disease implications. I can't talk about that. I probably can't talk about epigenetics in general without talking about the Dutch famine, which was a terrible thing that happened in the, I believe, 1944, where it was a combination of a food embargo and a harsh winter and war times that just caused this country, that caused the Netherlands to fall into a famine. Uh, it was pretty severe. I was reading about how, and this is not a joke. I'm not making a joke, but they would eat tulip bulbs. I mean, there was nothing to eat. And there have been a lot of studies looking at how people who were pregnant at the time, how that potentially affected later generations.
She's giving birth to a slug.
Oh, I don't like that. Don't say that. That's a baby.
Is it a baby?
It is a baby.
Maybe
Why?
the screen is. Let me look at my own screen.
Why would it be a slug?
Oh, the feet are up, of course. I thought it was like ears, little eyes.
Yeah, see the ADHD memory thing, because there's the prefrontal cortex, which is also why there's that trouble with executive functioning. If you have like impaired methylation in that area, potentially people with ADHD have that, then you can't. You got goldfish memory. Dude, I would like how that would just sound so terrible. Like it's like, oh my god, I want to eat a whole chocolate truffle tower cake, but all I have is half a green onion. So terrible. So those starving mothers, if you are not getting enough nutrition, you are affecting the environment for the fetus in utero. I'm eating them. And so if you're malnourished, the fetus is going to be malnourished too. The in utero environment itself is going to be malnourished, which will involve epigenetic changes in the developing fetus. That could either be, this is my own imagination. Jesus. I can imagine those epigenetic changes being to compensate potentially for the lack of nutrition, which could come into play in later generations because it's been shown actually that uh, what was it? I think it was children of mothers who were pregnant during this time were at increased risk for diabetes and obesity, showing that the genes that were subject to epigenetic change had to do with hormones and metabolism. And then, even farther down the line, there were a lot of um, children who were affected, like the grandchildren of these people, because the gametes that formed in utero of the person living in the famine were also subject to these epigenetic changes. And so they were then directly contributed to the grandchildren. They are made from those, I don't want to say famine gametes. That sounds extreme. But yes. So when epigenetic changes are persisting through generations that are being made from those gametes that were in that in utero environment, we call that intergenerational epigenetic inheritance. And if it continues to persist, and those who were not subject to famine environment, including in utero famine environment, that are still experiencing these epigenetic changes, we call that transgenerational epigenetic inheritance. So it seems like, I mean, those epigenetic changes really did persist through a lot of generations of Dutch people, and not just to like the children who were actually like fetuses at the time. To be your famine granny. An impression of Amy ordering food at her favorite restaurant. Yes, I can do that right now. Yep.
I can't wait.
All right, here we go. I'm not even doing it yet. Can I have another minute? And then they come back. Oh, I didn't read it yet. And that's it. Famine granny, famine granny, famine granny. There was more than just cardiovascular issues and obesity. Oh yeah, the lungs. Higher instances of mental health issues. And these, of course, were pretty pervasive beyond the generation that lived through the famine. I hate you guys. Yeah, even if it's like a page, she's like, oops, I'm not reading it. I'm looking at it and I'm not reading it.
What's the Egyptian god that like weighs your soul against the feather or something like that?
Obama?
Shut up!
I don't know, ISIS.
ISIS.
Anubis! It's Anubis.
Anubis.
The
Yeah,
dog.
I'm Anubis, like considering different like dishes to eat for dinner against us, the weight of the soul.
I cannot believe you compared yourself to the Egyptian god of judgment who has to make the most important choice in the world instantaneously.
Yeah.
Because that's the opposite of you.
Well, guess what? I'm a puppy, so it matches. She looked away from me. She looked away from me.
Sleep is rampant. Sleep issues are rampant. I hate you. You're making my glucocorticoid receptors upregulate. I hate you. Okay. We all love sleep. Everyone who would come to buy weed for me at the dispensary who was mean to me was buying it because they needed to sleep. So, it is cortisol #1 enemy. Absolutely. And dude, the glucocorticoid receptor is gonna be in pretty much all of the the other diseases that I'm going to talk about, because it's the best studied gene, of course, but also it's heavily influenced and influences our stress response. So, and cortisol is the glucocorticoid. Is the glucocorticoid that binds that receptor. So, in utero, if if the fetus carrier has high levels of stress and high levels of cortisol, then that is going to lead to epigenetic changes within the fetus that are going to upregulate the glucocorticoid receptor in the brain. And when that happens, that can make it so that the stress response is a little extra, like it's hyperactive. It's more than it needs to be. And of course, that can contribute to things like insomnia. Sufficient parental affection, interestingly, is associated with healthy stress responses and therefore a normal amount of these receptors. So, much like epigenetic changes can cause sleep issues can cause epigenetic changes. So, sleep deprivation can affect genes that are related to just our normal functionality. So, anyone who has been sleep deprived feels this pretty heavily. But that can be in genes related to, again, hormones and metabolism, which leads to higher instances of obesity and high blood pressure. So again, these are changes that are being made within you that can change within the span of a life. Like, they're not changes that are made to your code, to your DNA. They happen and then you have the change. But being sleep deprived, for example, if you are changing the epigenetic patterns in these hormone or these metabolic genes, then you are in real time increasing your chances of having high blood pressure, if that makes sense. You know, it's something. This is what's so interesting about epigenetic changes and why people put a lot of stock into them and want to study them so much is they're changes to our gene expression that can just be like, okay, now you have high blood pressure because you didn't sleep good, because you downregulated genes that help you regulate your blood pressure, you upregulated genes that give you high blood pressure. I mean, it's real time. It's like real time affecting your genome, but not the code. Oh, and cognitive deficits. How could I forget that? Sleep deprivation can cause increased methylation in genes that are involved with, like, I don't know, helping you keep a nice circadian rhythm, which really means that, I mean, that's like it's a vicious cycle, right? And even though these changes can be reversible, pretty hard to reverse. You probably get that oh, I thought that was onion, but it was my shadow. You probably get that floaty feeling because your body is using resources to keep you, like, energized. Like, your body is depleting your resources. And so I would say that probably you feel floaty because you're being hollowed out internally. Maybe, possibly. All right, everyone's favorite, mental health. The methylation patterns, the epigenetic patterns in the adult brain can be heavily affected by uterine environment, childhood treatment, trauma, stress, all that jazz. No, no. See, these are like those pseudoscientific fucking fucking hawks that come in and draw these conclusions, and then they have Instagram ads that are like, I know the secret to getting skinny, and it's not sleeping. Like, fucking hate them. um, there are lots of activities in your brain, like memory and learning, that require epigenetic changes. So stuff like this, due to environment, childhood trauma, can act to dysregulate those things that you want to keep normal and regular. There are still the glucocorticoid receptor. This is such a big one. It's a critical mediator of the stress response, and it's subject to all these epigenetic changes. People who have a lot of childhood trauma almost always have high glucocorticoid expression in their brain. And the reason they know this is because a lot of these people, these are autopsies. So I'll let you guys fill in the blanks. Oh, sorry, decreased. Wait, that doesn't make sense. Okay, they're talking about a specific area of the brain. Either way, dysregulated glucocorticoid. There's also there's a protein called brain-derived neurotrophic factor, which is decreased in depression. It's another protein that you need for normal brain cognitive function, and it's just been noticed that again, I don't want to say how these results have come about. I'm just gonna say that these are autopsies. And then another big one is COMT, catechol O methyltransferase. Not that kind of methyltransferase, but COMT is an enzyme that breaks down dopamine, and it's been seen that people with mood disorders have a lot of COMT. So constantly breaking down dopamine means that you don't have a lot of it, and that you can't utilize it for a lot of cognitive functions like emotional regulation. And again, these specific ones that I was reading about have to do more with like really early exposure to really stressful stuff. There could be explanations that are more like things that happen throughout life, like my example that I was talking about with sleep deprivation, where it's like a more real-time like thing that can happen. I'm sure that there are examples of that for like mental disorders. I mean, sometimes you can just have something happen to you, and now you have a mental disorder. So depending on the stress or the trauma, there are really serious epigenetic changes that can take place in certain areas of your brain that can bring about a disease state. So, ending on a really happy note here. Oh man. You guys did it. You guys did it. Watching the Animaniacs gave you ADHD. Dude, it's like 3 characters on screen, 3 different plot lines happening in one whole scene. Also, I can never get fingerprints out of my head. I'll take questions, and also, didn't I have a... Oh yeah, my explanation was why we always transcribe from the exact same strand of DNA every single time. It's always the same one. Our body does the same one. And that is because DNA has direction. One strand goes one way and the other strand goes the other way. They are anti-parallel to the other, and we know that because we have the 5' and 3' ends. When we are transcribing DNA and we're like going along and adding the nucleotides to the adding the nucleotides to the RNA strand, we can only do that in one way, and my brain is trying to make sure I'm saying it right. 5 prime to 3 prime. You have to start from the shit. You have to start from the 5 prime end of the DNA because wait. I need to look at a picture of DNA really quick. Basically the enzyme that makes RNA can only add it in one direction because chemically speaking, you can't add a nucleotide to I think the 3 prime end or the 5 prime end, sorry, because it has a phosphate and the phosphate is already happy. Oh shit, it just crashed. It didn't like that. You can't add bases in the other direction because I will show you when my thing reloads, but phosphate is already happy because it has all these oxygens attached to it. So it's like it doesn't need um when molecules are happy, they're happy. They don't want anything to be added to them. And that's the case of the phosphate. But the 3 prime end, which just has an OH, you can add stuff to it just fine. Which is why you can't do it on the other strand of DNA, because otherwise you would just be trying really hard to shove it onto the phosphate that doesn't want any more. Ha! Okay. Here's the phosphate. Here's the OH. That's the 3 prime end. This used to be an OH. And then we were like, what if we added a cytosine because we're making RNA? And then boom, you can do it because OH is like, whatever, dude, I'll become water. It's totally fine. But you can't do it the other way around. ADHD were the direct market for Animaniacs. Honestly?
I was there.
Yeah.
I saw it happen.
Honestly true. Yeah. This is for the kids who don't want to watch more than a millisecond of the same thing. Yes, everybody has PhD now in epigenetics. And
Money, please.
money, please. And I thank you all heavily for joining. This was great. This was my... I think this might have been longer than Sonic Hedgehog, which I didn't expect because Sonic Hedgehog had a lot of shit. There was a lot going on in that motherfucking shit. But yeah, I'll put the vote up tomorrow. Obviously I've just been like recycling like things that people have, like the thing that's gotten second place in the vote, I always add that back in. But and or I just think about other stuff that might be fun to talk about, but if you guys have ideas, I will take them and I will put them in the vote. Unless it's like, how is poop made, in which case I don't, maybe not that.
Rebecca, how's poop?
No.
How about you tell me after the show?
Poop is made in the balls. Poop is made in the balls. There's nothing else to it. See, shortest lecture ever.
Speed run. Run quick for speed running.
Speed running poop.
Call that diarrheas.
Diarrhea. Ooh, I can do those. Those, they're very difficult and abstract, but I think they have enough of a molecular basis that I can bridge the gap. Especially dreams. I've read about dreams before, and that shit is cool. So I can get that shit in there. And memories would be good because I really only touched on it here. Like, if I did more research, I could probably really put together a full on, like, more bridge crossing between abstract molecular things and, like, what my brain do when I'm trying to write a book. They really are. They're how come the brain doesn't understand itself? Because if you do a lot of research in like if you look into even anatomy, anatomy's easy. We just that's this and that's this. There's so much discourse and so much controversy around what we name our parts of the brain. It shouldn't be the basal ganglia, it should be the basal nuclei because it's inside the central nervous system, not the peripheral nervous system. Also, the amygdala isn't a part of the basal ganglia. Yes, it is. No, it's not. It's fucking nuts. Well, technically, this isn't Brodmann area, peepity poopity. It's it's very passive aggressive and hilarious. Thanks again, guys. This was great. You guys are the best. Look out for the vote tomorrow in the Discord. Okay, I love you. Goodbye.